Passive transport is how cells move substances across their membranes without using energy. Today, we'll focus on three key types of passive transport: simple diffusion, facilitated diffusion, and osmosis. These processes are crucial for maintaining cellular balance, absorbing nutrients, and removing waste. Let's break down how each of these mechanisms works. First, let's explore simple diffusion. In this process, molecules move directly across the phospholipid bilayer of the plasma membrane, from an area of high concentration to an area of low concentration. This movement happens due to the natural kinetic energy of the molecules and continues until equilibrium is reached, where the concentration of the molecules is equal on both sides of the membrane. Since it's a passive process, simple diffusion doesn't require any energy input from the cell. Molecules that typically move by simple diffusion are small, nonpolar, and lipid-soluble. Examples include gases like oxygen and carbon dioxide, as well as small, lipophilic molecules like steroid hormones. Oxygen moves into cells by simple diffusion, going from higher concentrations outside the cell to lower concentrations inside. Carbon dioxide, a waste product of respiration, diffuses out of cells to the surrounding environment. This simple yet vital process allows gases and other small molecules to move freely across the membrane, maintaining essential processes like respiration. Next, we have facilitated diffusion, which is also a form of passive transport, but it involves the help of transport proteins. While small, nonpolar molecules can diffuse directly across the membrane, larger, polar, or charged molecules need assistance. Facilitated diffusion uses two types of proteins: channel proteins and carrier proteins. Channel proteins create water-filled pores that allow specific molecules or ions to pass through. One example is aquaporins, which are specialized channels for water molecules. These proteins facilitate the rapid movement of water into and out of a cell, playing an important role in balancing water levels, especially in cells like red blood cells or kidney cells. Carrier proteins bind to the molecule they transport, undergo a shape change, and then release the molecule on the other side of the membrane. A common example is the glucose transporter, which helps glucose move into cells. Since glucose is too large and polar to cross the lipid bilayer on its own, it relies on carrier proteins to enter cells through facilitated diffusion. Although facilitated diffusion uses proteins to assist, it still does not require energy and relies on the concentration gradient, just like simple diffusion. The third type of passive transport is osmosis, a specific form of facilitated diffusion that involves the movement of water across a selectively permeable membrane. Osmosis occurs when water moves from an area of low solute concentration to an area of high solute concentration, aiming to balance the concentration of solutes on both sides of the membrane. Water molecules move through aquaporins, which we mentioned earlier as specialized channels for water. Osmosis is critical in maintaining osmotic balance in cells. For example, in animal cells, osmosis helps regulate cell volume. Too much water entering the cell can cause it to swell, while too much water leaving can cause it to shrink. In plant cells, osmosis maintains turgor pressure, which is the pressure of water inside the cell pushing against the cell wall, helping the plant stay upright. Osmosis is crucial for maintaining water balance in cells. and it's controlled by the tonicity of the surrounding solution. Tonicity refers to the concentration of solutes in the extracellular environment relative to the inside of the cell, and it affects the direction of water movement. Let's explore the three types of tonicity. Isotonic solutions. In an isotonic solution, the concentration of solutes is the same inside and outside the cell, meaning that water moves in and out at the same rate. Cells maintain their normal shape because there's no net movement of water. This is the ideal environment for most animal cells. Hypotonic solutions. In a hypotonic solution, the solute concentration outside the cell is lower than inside the cell. This causes water to enter the cell by osmosis, potentially leading to swelling or even bursting in animal cells. In plant cells, however, a hypotonic solution increases turgor pressure, which helps maintain the plant's rigidity. Hypertonic solutions. In a hypertonic solution, the solute concentration is higher outside the cell than inside. As a result, water leaves the cell, causing it to shrink or become crinated. In plant cells, the loss of water leads to plasmolysis, where the cell membrane pulls away from the cell wall, causing wilting. Understanding these environments is essential for maintaining osmotic balance in cells and ensuring they function properly. All three processes, simple diffusion, facilitated diffusion, and osmosis, share a key characteristic. They are passive. This means no energy is required, and molecules move down their concentration gradients. Simple diffusion allows small, nonpolar molecules to move freely across the membrane. Facilitated diffusion uses channel or carrier proteins to help larger or polar molecules like glucose and water pass through. Osmosis is the movement of water across a membrane, balancing solute concentrations in and out of the cells. These passive transport mechanisms play a fundamental role in cellular processes like nutrient absorption, waste removal, and maintaining water balance. By allowing substances to move freely across the membrane in response to concentration gradients, cells can efficiently regulate their internal environment without expending energy.